but you do not have a lot of information regarding this. So whatever I feel is something similar to whatever has been given in your textbook, I will send. Otherwise, excuse me. Otherwise, please follow your textbook. That's got the most amount of information regarding the same, basically, regarding the protocol for making slides and the different kinds of techniques. And then we have already seen, so I'm not exactly going to go through the same again, the staining methods. So we have seen how to do the Q banding, the R banding, the T banding, and the G and the C banding. If possible, I'll just like try to share the same with you, and tell you the same thing basically. So when it comes to the Q banding and the different kinds of banding techniques, you get to see that over here you have chromosomes in the Q banding. They are stained with fluorescent dyes such as the quinacridine. Uh, just taking. So you get to see all of this in the karyotyping. So when it comes to the R banding, it's generally the reverse of the Q banding, wherein you have the fluorescent as well as the non-fluorescent staining methods that are there. Right. Share my screen with you so that you'll understand what I'm talking about. So already this is there with you, so I'm not going to go into much of the details as such. So you can get to see in the Q banding, you have quinacridine, which is the dye that is used. And in this, what you do is we have given the protocol for the same, basically. Dehydrate the slides by dipping in alcohol, and then you decrease the concentration basically from 90 to 70 and 50 for around 1 minute each. Rinse or wash in the distilled water, and then you wash the slide in a phosphate buffer, then stain with the quinacridine mustard and or the quinacridine dihydrochloride, which is around 5%, and then you rinse in phosphate buffer and mount and watch under or see under the fluorescent slides. So Q banding stained over here, you get to see dark and light stains that occur. So dark stains are rich in AT bases, light stains are rich in GC bases, and you can see the heterochromatin and the euchromatin region. When it comes to the G banding, it is the GIMSA banding, wherein you have the chromosome. Again, it is treated with weak trypsin or different kinds of enzymes such as the urea or proteases. Then GIMSA is used. Banding again, you get to see that you treat it in normal and saline, and then trypsin 0.25% or 25% solution, 0.25% solution for around 10 to 15 seconds. Then immerse it in 70% ethanol, stained with GIMSA which is around 10%, and that is for around 6 to 10%, minutes. So again, in this, you get to have in this GIMSA banding, you have the cell concentration that is given, and you have different kinds of in your textbook, at least, the different kind of cell source of the trypsin is given, the age of the overnight slides is given, and the cell concentration. According to that, you're going to get the G bands available. So for optimum G banding, you have around you have to make the slides. You can watch under or see under the phase contrast microscope, basically. And then for high resolution chromosome banding, you see that you have this use of elongated methods to obtain a high percentage of the prophase and the pro-metaphase spreads. They are the chromosomes are actually less condensed in when as compared to the metaphase analysis, and number of identifiable bands is also more, in fact, so that you have a more sensitive karyotype that is available. So you have the R banding or the reverse banding over here. I'll see that. think I have R banding here, right? Never mind. You just please listen to. So in the R banding, what happens is a differential staining of the chromosome so that you get the R bands is done. It is the reverse of the Q banding, in fact. And then you get the G and Q banding. Darkly stained R bands are basically G and Q light bands. So whatever euchromatin and heterochromatin gets stained dark and light, respectively, which is the reverse in as seen in the G and the Q banding. You also have DAPI banding, wherein it is the preservation of the chromosome morphology, basically, which depends upon the three factors, that is, type of aging, pepsin treatment, and denaturing technique. So, dry heat aging provides a sharper DAPI bands, but decreased hybridization signals compared with the chemical aging. So, you have different kinds of chromosomes that can be denatured while preserving their architecture, that is the structure of it. So you have different kinds of chromosome spreading that can be done, followed by use short aging process. Then you treat it with protease, and then you gradual denature it using the hybridization. So this gives you a what do you call it? The DAPI bands pictures. I just happen to have the images of it. So let me see if I can share with you. Not much information has been given or is there regarding the same thing. I tried to actually check whether it's possible. You have positive, negative staining. All of this is there, but this is not good. No, no, sorry, not this one. This is not the slide that I was looking for. Let's see. Anyway, please have a look into your textbook. You can see the DAPI banding and its image basically, and which shows, using a hybridization buffer having around 12 to 5% of dextran sulfate and different forms of the formamide that is present, then and the process among it. And then you get to see that, use of dextran sulfate during the denaturing process increases the chromosome thickness, altering its structure. At the same time, formamide brand influences the process so that chromosomes either are very thick or they're distorted with having uneven surfaces. So that exactly, they have just kind of showed with different kinds of bands how we are going to get the chromosome pictures basically. So when it comes to the fluorescent banding, you get to see that staining the chromosomes with fluorochromes, that is those fluorescent label dyes such as the acridine orange or ethidium bromide or propydium bromide or DAPI, that is 4,6-imidono 2-phenyl indole or FITC, which will actually help us. So hybridization of a probe directly to the chromosome, then it has become extremely common basically. And with the availability of the nucleic acid probes also, this you can radioactively label the chromosomes and then, view it under the fluorescent microscope basically. And you're going to get a very good picture of the different kinds of chromosomes, their structure for the karyotype. So you have chromosome painting also over here. See, I have that particular slide. So you're actually going to get something of this sort after, your metaphase chromosomes after banding and staining. Then you can get to see that you release the chromosomes from the cell, and after staining, you're going to see the hybridization buffer or the different kinds of buffers. And once you're staining them in the GMSA dye or inside any other dye, quinacridate dye for that matter, or using DAPI and reverse banding and all that, you're going to get this kind of image. So this is the human male G bands, that is the GMSA band, with the smear. This is the human karyotype that you're going to get. So abnormal results or the genetic conditions can be detected with the same. Right. I'm having an issue with my cursor. I'm not able to for the next slide altogether. Please give me a moment. Right. In the case of chromosome painting, like I said, you have fluorescent dyes. Like I had already mentioned, you can use the fluorescence in situ hybridization also. You're going to get the picture of something of this sort. And then with this, the technique is used is increasingly sophisticated. Basically, many probes are used at the same time. That is why you're getting a beautiful color picture actually. Right. So fish techniques have also been used in this so that traditional chromosome analysis can be advanced as such. And then you have in this fluorescence in situ hybridization technique, what happens is complex or small cytogenic rearrangements which are not exactly you're able to see in the others, you can actually use using this fish. It also allows the rapid screening of large number of the non-dividing cells also at the same time. So you have three kind of techniques that are involved in the fluorescence in situ hybridization. One is the cytogenetics. You have the fluorescence microscopy and the DNA hybridization, all of which put together you have the fluorescence in situ hybridization technique. So many different kinds of fish probes are available. Then you also get to see that alpha satellite probes have the DNA which is isolated from the centromeric region, and this is unique for each and every chromosome. It can be used as a probe, and they're used to detect the numerical chromosomal change. So used in detecting the genetic abnormalities as such. So you can see the sex chromosome content, presence or absence of individual chromosomes, and nature and the extent of the large structural abnormalities. So they've given different. I think you already have this particular slide, so I'm not going to go through it again. So when it comes to microscopy and imaging, you get to see that my video, I don't know, something is wrong somewhere. Please give me a moment. I'm trying to close the PowerPoint and right. So let us see the next topic that is there in our textbook, which is regarding microscopy. So when it comes to microscopy, I'm just going to go through it basic, in a basic manner, what exactly is a microscope and how it has been used, and different kinds of, microscopes are there. A brief history of a microscope is also there, basically. I guess you're able to see the screen right now, right? So when you get to see a brief introduction of the microscope, you get to see that micros means small, scopy means to look at. So it's an instrument used for seeing objects that are too small that cannot be seen with the normal eye. So the science of investigating the small objects is known as the microscopy. So historical background, if you come to it, you have just seen basically that Hans Jensen and Zacharias Janssen developed the first microscope. Then in 1609, Galileo Galilei had developed the compound microscope. 1620, Christian Huygens, another Dutch person, developed a ocular system that can be corrected. So Anton van Leeuwenhoek and Robert Hooke played a very important role, especially in the field of microscopy as well as the field of biology. You all know that. So again, what are the variables used in the microscopy? You get to see there are certain words that you get to listen to in microscopy. So the first one is magnification. Please give me a yes. So magnification means the degree of enlargement. Number of times length, breadth, or diameter of an object is multiplied. Again, this depends upon the optical tube length and your focal length of the objective, and you have a magnifying power for the eyepiece. So a total magnification is magnification of the eyepiece into magnification of the objective. To go through with this. Resolution is the ability to see how closely adjacent the details, the structural details are separate and clear, that is distinct. So you have a resolution limit or the limit of resolution, which is a minimum distance between two visible bodies that you can see separately and not in contact with each other. So they have given a value based on the kind which is there basically 0.61 into W, that is the wavelength, lambda by numerical aperture. Numerical aperture is the ratio of the diameter of the lens to the focal length, which is around n sine theta by 2. So n is the refractive index over here. And theta is the angle of capture from where you're seeing that particular angle. So capacity of an objective to render the outline of the image clearly and distinctly is known as the numerical aperture. Aberration is again, wherein you get to see the difference. Spherical aberration and chromatic aberration. So you have see the object where it is and where you are able to see is the difference of the the difference or the distance that you can get to see is known as the aberration. So for chromatic aberration, you get to see that it is of two types aberration, that is spherical and chromatic. So the image where it is formed and the distance of it is known as the aberration. So you have different kinds of the definition also. The incident light when it falls. This is the focus, basically the focus and the red focus. This is how the incident light falls, and you can get to see the difference in them. This is the chromatic aberration. When it comes to the working distance, you can get to see that this is the objective, the distance between the front surface of the lens and the surface of the cover glass or the specimen is known as the working distance. Contrast is differences in the intensity. So you have different kinds of intensity, and you can use a contrasting dye for that matter, or the light where it is present. All of this makes a difference in your viewing of the object. Different kinds of properties of the microscopic objectives are there. So when it comes to that, you can get to see basically that the microscope has a lens which is having different kinds of magnification. So different kinds of characteristics are there basically. When it comes to the lens, like a basic microscope has the foot, the limb, the tube. See, this is a compound light microscope. This is the foot basically. And you can get to see that this is the limb or the arm as it is called. Then you have a tube and you have the objectives. So these are the objective lenses over here. And you have the eyepiece. This is the eyepiece or the ocular. This is the fine focus control. Basically, you can find focus control and the coarse focus adjustment knob. This is the stage adjuster that is there. And you have a resolving nose piece. You have a revolving nose piece also. This is a revolving nose piece. You have a mirror also, and you have a substage condenser or a condenser as it is called, and you have a condenser iris diaphragm, and you have a swing out filter ring that is present, and you have a light control or the light intensity control. So this condenser focuses control the and if this is not exactly visible over there because it's on the other side, and it's approximately on the same level as the fine focus in the picture. Now you have a light source also over here. And these are the lenses that are there. So to get a depth in the field, any lens focus, you'll have a plus and a minus and a certain minimum range that is present. This can focus without any kind of adjustment is known as the depth of the field. And for high power oil lenses, it is not exactly possible. It can only be increased by closing the condenser iris diaphragm and then reducing the working numerical aperture, but resolution is compromised. So working distance of the lens is the distance between the lowest point of the objective and the object in focus. So you have different kinds of magnification also in the lens. So the lens magnification is determined by the tube length, and that is the distance from the rear focal plane, and the focal length, distance from the surface of the lens to the point of the convergence of the rays. So you have different kinds of formulae like I told you. And like I told you, it is 0.61 by numerical lambda by numerical aperture. So tube length is usually 160 millimeters. So that is why you get the number so the focal length of the standard 10x objective would be 160 by 10, that is 16 millimeters, or 40x. In case of the magnification, would be like 160 by 4, that is 40x. Achromatic oil immersion lenses have focal lenses about 1.8 to 2 millimeters. You have different kinds of lenses, like I told you, afocal, parfocal lenses, high power, low power, and oil immersion. So again, numerical aperture changes, and numerical aperture is the number that expresses the ability of the lens to resolve the fine detail that is present, which are observing basically. And you have different kinds of lens marking also. As you can see, the magnification 4x, 10x, 40 to 45x, and 90 to 100x. So is the kind of magnification that can be seen. And then what do you call it? Numerical aperture, distance is also being given, and approximate focal length 40 millimeters, 16, 4 millimeters, 1.8 to 2 millimeters, like I told you. The working distance is also being given. 17 to 20 millimeters, 4 to 8 millimeters, 0.5 to 0.7 millimeters, and 0.1 millimeters. So that accordingly, the resolving power with light of wavelength in the given wavelength of 450 nanometers, that is blue light, it's given. For scanning the microscope, it is around 2.3 micrometers. For objective, it is 0.9. High power, it is 0.35. And oil immersion, it is 0.18 micrometers. So that's what. And for lens marking also, you have different kinds of lens markings that are there basically. And the lens marking is having certain engravings on the objective lens. So magnification either as a whole number or as a ratio, that is 100 or 100:1. Then tube length in millimeters is given. And then you have cover slip designations also that has to be seen, which can be seen after the tube length. So 17 means cover glass of around 0.17 millimeters, and plus 0.01 should be 0. So if at all there is no covered slip, then numerical aperture is under the magnification, which is marked. Example 1.3, like that. And correction can also be done with which is given as, for example, in your textbook, you can just have a look at it. 170 by 0. So you have 17 and that number has been given. So this means that it is a 170 millimeters tube. Cover glass thickness is 0.17 millimeters, and it is a piano lens, PI or PL. Piano lens, flat field and having an apochromat oil immersion lens which has been corrected for 3 different colors that magnifies 100 times, 100 by 1.32, and the numerical aperture is 1.32 over here when it is under oil immersion. So oil immersion, usually it is cedar wood oil that you keep using usually, and other markings are also there. So when it comes to the different objectives, you can get to see that magnification is under 2 stages, that is the objectives and the eyepiece with the magnification that is there. So which ranges, the magnification ranges from 15x to 1500x. Again, binocular tubes have own magnification factor ranging from 1.25x to 1.1 is to 5x. So all of this has to be taken into account while calculation. And so if at all the 10x, 20x, 40x, they're all known as the dry objectives, meaning air is present in between the lens and the slide. Now in 100x, that is the oil immersion objective, you place a small drop of oil inside the slide in front of the lens of the objective for viewing. Reason is that lens is oil to the slide, so it will become clear later and you can see at a higher magnification. So optical construction of the objectives also for this work gives a good image, good quality image from around 4 to 6 lenses of different shapes and kinds. So now you have 2 other additional numbers that are engraved on the objective mount. The tube length, which is 160 millimeters, cover glass thickness is around 0.17 millimeters. So good objective lenses are necessary for any kind of microscopy to have. So you have achromatic objectives and apochromatic objectives over here. So when it comes to the achromatic objectives, they are not exactly very expensive. You can correct for 2 colors and you can see all the different colors with different wavelengths that is available in the visible light basically to focus, and you can see the image. They usually are held or they're designed in such a manner so that they perform the best with the violet light filter corrected. Whereas in apochromatic objectives, they're superior to achromatics. And what happens is you get instead of getting the 2 wavelengths, your blue light and the red light wavelength to come into a common focus, you have or you correct 3 or 4 wavelengths altogether, and then you're going to have a higher numerical aperture. This gives a better resolution than the achromats, so they're much more expensive. You also have semi-apochromatic or fluorite objectives also, which are used, which are intermediate between the achromats and the apochromats, and you have the neofluor objectives for this case. You also can use quartz lenses and the lenses for fluorescence. When it comes to the eyepiece also, you have different kinds of, it is made up of, see, the primary image that is present is magnified by the eyepiece or the ocular. So this is again made up of 2 lenses, which is known as the field lens and the eye lens. This is the ocular or the eyepiece. So calibration of it, they have just kind of shown basically. So when it comes to measuring, we'll come to that. But anyway, so different kinds of eyepieces also are there basically. And they come in different magnifications such as 8x, 10x or 12.5x version. Some of them have even for example, this oil immersion 100x lens has this numerical aperture of 1.25 eyepiece. This gets more magnification basically, so that you can get to see what do you call it? What happens is when the oil immersion lens is used with a numerical aperture of 1.25 magnification, it should not be used for more than 1250 magnification, because this will distort the image. And markings found on eyepieces include this magnification of around 8x to 1.25, 12.5x, etc. And then you also have a symbol appearing like a pair of eyeglasses for glass wearers. And then you also have that, oculars were corrected for such kind of aberrations in the objectives. So this makes it important for the oculars and objectives to be coming from the same manufacturer. If the manufacturer differs, then you're not going to exactly have, or there is going to be a difference in your eyepiece and your objective power basically. So you now have infinity corrected lenses that are now available, which helps correct this issue. So you have the high-gain eyepiece as well as different other modern designs basically in the eyepieces. And you also have high point eyepieces, which are the wherein, if you have glasses also, they can be kept on. Otherwise, you have an issue with having the glasses and then looking into the eyepiece. That will give you a aberration basically. You're not exactly able to see the image properly. You now have the condenser over here. In the case of the condenser, the function is to fill the objective with a complete light. So it helps in condensing the light so that you can get to see the image properly. So they also have a numerical aperture engraved on the mount. Typical value for the condenser's numerical aperture is 1.2. This can be attained only when the condenser is oiled. For different kinds of magnifications, it might become difficult difficult to illuminate the field of view entirely with a condenser which is around 0.09 or more basically. So again, you have different kinds of condensers over here for low magnification, for high magnification. So you have what do you call it, ebb type condensers which are one of the most or the cheapest variety that is there. And then you have the aplanatic condensers, which is slightly less expensive than the top range, which is the achromatic aplanatic condensers in fact. And then you have condensers based on the kind of field of light, that is illumination. So you have a dark field condenser, you have a phase contrast condenser, and then you have an interference contrast condenser. In the case of dark field condenser, this illuminates the object from the side instead of straight through in the bright field. So background is black, therefore objects are black. Classic example is when the curtain rooms are drawn, except for a narrow slit, the tiny dust particles become visible in that beam of light. So this is how a dark field condenser works. Phase contrast is when the what happens is it is the simplest of the type that is present in fact. So you have a ring-shaped annular stops, one for each phase contrast objective. You have a mounted and a centrable rotating disc, as you can see over here. And there is also a clear position which allows for normal observation. Okay. And then you have the interference contrast condensers used in conjunction with additional special components in the microscope. You have this is the mechanical stage. Again, they have just given different methods, it can move horizontally, it can move vertically, so that you can, adjust and view your image as such. So then vernier scales are present on the mechanical stage over here, so that, if 0 on the vernier scale coincides with the mark on the stage scale, then the whole number indicates. Then if the 0 lies between the marks on the stage scale, you can read the next lower whole number and the tenths of it, determining the vernier's opposite division on the stage scale. So some stages are removable as well. Scale should not be moved when the stage is removed, and it should be professionally cleaned. And this is vernier scale. All of it gives you the size of the object that you're looking at. And then you have different kinds of accessories again, a rotating stage that is present, and based on the convenience. So you have a lot of other equipment that is allowing you to make or help look the image properly. So And then when it comes to the image quality, it is assessed for the by the following basically. One is brightness, second is focus, third is resolution. So how light or dark is the image basically, and can you adjust the brightness? So the numerical aperture is also dependent upon the brightness, because the numerical aperture of the objective lens is longer than the numerical aperture. So you get a brighter image basically. Focus, you need to see it properly. Is focus, if you're able to adjust the length, and if you're able to adjust, the magnification, are you able to see the object clearly and distinctly? So this can be controlled by the focal knobs, and the thickness of the glass cover, and also the correct over glass thickness is also written on the side of the objective lens. This will help you in focusing and seeing an image clearly. And when it comes to resolution, you can see that how close are the 2 points that are present in the image, and how before they're no longer seen as separate entities. So resolution is again related to the numerical aperture of the objective lens and the wavelength of the light that is passing through the lens. So if the wavelength is short, you have got a better resolution. If the numerical aperture of the objective lens is high, then you have a good resolution. So these make a difference, or how better you can get to see an image. Also, you can get to see the contrast, difference in the lighting areas, and how exactly are you able to see the the whatever you're watching inside. For example, pollen grains, or for that matter, any kind of fungi or the mycelium. So when you're looking at it under the microscope, how clearly are you able to see using the intensity of the light and the diaphragm or the pinhole aperture? And if at all, you have any kind of chemical stains applied, how clearly are you able to see the specimens? Now you have digital imaging. So with the digital imaging, you have a sensing device, which is you uses the voltage to form the image analogous to the brightness, and color also it can be adjusted, and then you can store it as a grayscale image. So another type of the camera that is used is a solid state sensor, so that you have a charged coupled device. It's known as a CCD camera. They can collect the charge over the time, and then they're used for recording the dim images, such as the fluorescent probe signals. So this all can be used to check the different kinds of images, basically the resolution, the contrast, how clearly you're able to see as such. When it again comes to contrast and resolution, it is that which is for example, it can get to see that phase contrast and interference contrast microscopes come into place. So contrast is the difference between the brightness of different details in an object. So as compared to the background, how different, how much of difference are you seeing as compared to a background? So black and white computer image, you'll have pixels in around 250 shades of gray, intense white to intense black. So this is known as the gray value. So if the values in the image are from around 30 to 50 only, that means that you cannot see properly. It's either very gray in color. So if the value ranges from 30 to 200, image will be contrasty. You're able to see a better black and white picture. Background has uniform pixel of 50, image pixel is around 70 to 150 or 120. Again, background pixels have a better value. You're not able to see properly. So similar thing you can apply to the images or whatever you see inside the microscope also. And when it comes to resolution, how accurately you are able to see an image. How accurately are you able to see the pollen grain? So resolving power is the highest resolution possible with the lenses that are available. So again, you can compare it with the pixel sizes that you get. So maximum resolving power, that is the optimal optical pixel size, is around 40x by numerical aperture around 0.65 objective. So you get it around 0.05 micrometers for a 100x numerical aperture 1.25 objective. It is around 0.25 microns or U. Whether that value is really you're able to get it depends upon the particular microscope, microscopist. So they have just given you low contrast or correct or low resolution pictures over here. And then apertures also make an important aperture is the hole basically, or how you look through the bright circle. You get to see that is the aperture, and this is different from the numerical aperture, which is a number basically. But they are both give you the image. Now, when you see the condenser iris diaphragm that is present over here, and you slowly close it, at the edge of it, you get to see that, for you need to refocus it for if at all you want a sharper image. So you have to center or decenter this condenser iris diaphragm basically, so as to get a good image. So you can see that take the eyepiece out, then the condenser iris is present. It cuts off the 15% of the aperture. Then you can get to see by examining it in different lenses, so that if you adjust the focus for a sharp image and see, you can get to see a better resolution of the image. So these are the things regarding the aperture as well. When it comes to the lens faults, you can see that there are certain issues that you have regarding the lens. So you take a 10x magnification of the lens over here and then focus it onto the slide. Then what happens is you get to see a shadow of the microscope. That is there without a built-in illumination. Now, when this condenser iris is fully opened over here, you get to see that there is a color change or a color fringe lights that are observed from red to blue as you vary the position of the condenser. So this is one thing which is which is the chromatic aberration. It will be severe for any kind of uncorrected condenser. So this aberration that we are seeing, spherical aberration and the chromatic aberration, is the lens faults that you're going to talk about. And in objectives, this degree of correction is also quite high. When you get to return to a normal observation and you see that the 10x objective, condenser iris and condenser position correctly are adjusted, you focus the object in the middle, then without changing the focus, you shift the object to the edge of the field view. Then again, you'll have to readjust the fine focus to see it in a better way, or that is sharper. So this is known as the field curvature. So this is one thing which is also there may be a small residual curvature that you can get to see. So these are the different kinds of lens faults. And again, for correct illumination, you get to see that you'll have to have a proper illumination as well, so that you get a very good or a better picture. You can see the object in focus, and the light source will also be in focus. So you have two varieties of the form of illumination. One is critical illumination and the second is the polar illumination. In critical illumination, filament of the light bulb itself is the light source and its focus is in the field of view. So image of the filament would be very objectionable. So the light bulb is slightly frosted, that is a little colored. Such an illuminator should have a collector lens to ensure the light from the illuminator forms a parallel beam. This means that the light source is present at infinity. All of this is optics. So you just, give it a reading for you to understand, in case you can, have any kind of videos that you can follow at, you can try and look at it so that you'll have a better understanding of all of these topics. So you have different kinds of illuminators with the light source at different positions, such as infinity, or at the same time, you have a lamp that is present at the infinity position, so that you can see the object at a better resolution, and there is no what do you call it, collector image that is formed. Now, when it comes to the color illumination, light source is the iris diaphragm in the lamp itself. So this is different from the condenser eye lens. This is the light source, right? This is different from the condenser iris. And its size can be adjusted so that only the field of view over here is illuminated. Again, you have light at the infinity instead of the critical illumination. So this is somewhat, it gives you a better contrast image, but it is slightly more complex to have to set up. Now, different kinds of, these are the parts of microscopes. You can see this is the calibration of the ocular microscope, the stage position, how to set it and to center it over the light source and calibrating it. And this specimen slide is positioned for measurement. You have the stage micrometer, you have the ocular micrometer, and this is how you're going to see the gradation. And this is how it is going to be appeared on the appearance of the stage microscope graduations. And then over here on the basis of the calibration, you can say about the total length of the yeast cell and its bud. So this is the length of the yeast cell and it's the bud that's coming out. Different types of microscopes are there again. When a light microscope and electron microscopes are two basic criteria that's there. And light microscope is wherein you can use it either in sunlight or artificial light. You have the bright field microscope, you have the dark field microscope, then you have the phase contrast and the fluorescence microscopes. Electron microscopes use electrons for illumination or for magnification. You have the transmission electron microscope and the scanning electron microscope. So bright field microscope is the basic microscope, produces a darker image against a bright background, has several objective lenses. Two types again, simple and compound. Simple has only a single magnification lens or magnifying lens. Compound has a series of lenses basically that is used for magnification with light passing through the specimen into the objective lens. So you use the oil immersion lens over here that helps in increasing the resolution. So you have either one or two ocular lenses. Resolution is up to 200 nanometers. So this is how you do. This is the glass cover slip without the immersion oil and with the immersion oil. Oil immersion is 100x magnification. Without the immersion oil, it is around 10x or 20x or 40x magnification. So this is the microscopic objective. These are the different kinds of lenses that are present. This is the glass cover slip that you add. This is the specimen over here, and this is the slide. So, and over the glass cover slip, this is the specimen, you have the immersion oil, which is usually cedarwood oil. So this is the kind of resolution. And then with the oil immersion, what happens is more number of rays are all falling onto the microscopic objective lens, because of which you can see better. Light, again, light source over here, condenser lens is over here. This is a specimen with slide on the top, and this is the objective lens over here. Projector lens is present. This is the eyepiece having the fine image. Advantages is using the light or stained images, and then you have the disadvantages again that staining may destroy the specimen as such. You get a low contrast biological specimen and resolution is limited to 200 nanometers. Again, for dark field microscope, again, image formed by the bright field microscopy is as such, the gram positive and the gram negative bacilli. When it comes to the dark field microscope, produces a bright image of the object against a dark background. Optical system is used to enhance the contrast of the sun-stained bodies as such. And then you have the Reinberg illumination, which is a setup similar to the dark field, but uses a series of filters, which is known as the optical staining of the system. So light waves pass through the dense structures within the specimen. They slow down compared to that pass through the less dense systems, basically. So you have an interference pattern. So they may show more dark areas and more light areas contrastingly as such. So in phase contrast microscope, this technique is best for seeing the live specimens such as the cultured cells. So the annular rings in the objective lens and the condenser separate the light. I have that. Just a second. Let me see. We'll finish the optics of the dark field microscopy over here. So this is the objective lens, this is the stage, this is the condenser lens, and this is the dark field stop. So and then you can get to see the different kinds of images of the like objects. This is Treponema pallidum, Leptospira, different species. Again, you need strong illumination that can damage the delicate samples. Basically, that is the disadvantage over here. When it comes to phase contrast microscope, it produces a high contrast image of transparent specimens. Advantages that you can see living cells. Again, principle is you have small, numerous rings basically in the objective lens and the condenser. They help in separation of the light. Light then passes through the central part of the light path. This travels around the periphery of the specimen. So interference produced by these two paths produces images in which the dark or the dense structures appear darker than the background. So this is the condenser or the annular. Wait a second. So you have the bacterium over here. This is the face plate that is present, and these are the deviated and the undeviated rays that actually help in cancellation out. So again, principle of the waves. So you have annular diaphragms and the face plates, which are very much necessary for the phase contrast microscope. So the face plate is placed at the back of the focal plane of the objective. It helps in increasing the phase difference by stopping the diffracted wave by 1/4 of the wavelength. This reduces the intensity of the direct rays. This equalizes it with the diffracted rays intensity altogether. So the waves get cancelled as such. So this is how a phase contrast microscope looks. This is the image plane. This is the condensers that are present over here. These are the annular condensers, as you can see. This is the face plate. This is the objective. This is where you get to place the specimen as such. These are the objective lens that you can see from where you can get. This is the observation, like ocular or the eye lens. Then these are the diffracted lights that they get cancelled with each other using the annular condenser and the face plate. So images of the phase contrast microscopy look like this. Advantages is you can visualize internal cellular components. You can differentiate between basic tumor cells and non-tumorous cells. So you can see the growth, behavior of the different kinds of living cells in a cell culture. So you can see or study the thin specimens that are present. Disadvantages is resolution is decreased because of the presence of the different kinds of aperture such as the annular rays. It's not exactly useful or ideal for thick specimens, and you have a shading off or a halo effect, shadow effect basically. That's there. When it comes to the fluorescence microscope, you can get to see that you are exposing the specimen to ultraviolet light or the blue lights. And then the specimens are stained with dyes such as the fluorochromes. So you have a bright image of the object resulting from the fluorescent light that is emitted by the specimen. So you have fluorochromes which absorb the UV rays. So they go to the higher energy. release that energy, and that you can basically see that the image because of it. The dye molecules return to the normal state, because of which there is a release in the excess energy. This is seen in the form of the visible light. This is known as your fluorescence. So what happens is this is the detector. You have a dichroic mirror that is present. This is the emission filter and the excitation filters that are present. This is the objective. And this is the specimen which is already dyed with fluorescent light. So the light emission of it, the excess, from the excess energy that comes out in the form of a light, the visible light. This is how you're going to get or detect the image. So you have different kinds of fluorescing dyes basically, and the kind of organisms that can be detected. You have auramine, rhodamine, so it gives out yellow fluorescence. You have acridine orange R, so you have an orange-red fluorescence in the presence of RNA, and yellow-green fluorescence with DNA. Then you have QBC, and you have different kinds of immunofluorescence also. So again, it uses this fluorescence microscope uses mercury or a xenon lamp. So this produces the UV light. The light comes into the microscope, hits a dichroic mirror. Mirror reflects one range of the wavelength, allows another to pass through. That is the function of the dichroic mirror over here. So this dichroic mirror reflects the UV light up to the specimen. Then UV light excites the fluorescence. UV light excites the fluorescence within the molecule in the specimen. Now, objective lens, whatever is over here, it collects the fluorescent wavelength light produced. This fluorescent light passes through the dichroic filter, and you have a barrier filter also. This eliminates the wavelengths, basically emission and excited filter that is present, so that you can see it to the eyepiece. Then this forms the image. So this is how the fluorescence microscopy works. And again, you have to maintain the microscope, give it to the professional who are able to make it in a who can treat it well basically, and who are able to see that there are no kinds of issues that come base. We cannot exactly just use or lubricate or grease without knowing what to do. And you have to see that you have to maintain the microscope very carefully, and you should see that whatever oil you're applying doesn't go into any other kind of inside the microscope basically. And then you have to clean that oil immersion front lens and all that by gently wiping them with a soft cloth or a lens tissue. And maintenance and cleaning of the microscope also makes quite a bit of impact on the image that you're viewing. And of course, if you're measuring any kind of, for example, pollen grain or yeast budding cells, or you're measuring the cells, there will be difference in the sizes according to the how the microscope is maintained also. And if you don't calibrate properly, you will definitely get a wrong image and wrong measurement as such. So to summarize this particular lesson, this electron microscope and all that, I guess, you already must have read it, but instead, I'm just there is this information. I will send this PowerPoint. You can just have a look at it. The kind of magnification in the electron microscope. You have both scanning and transmission electron microscope, and magnifications 10,000x, 200, 1 lakh x, basically time, the x means times. So that's what, and the optics of it has been given over here. Most basic ways wherein you can actually draw these images also, or just remember them. So I will share this with you later, once the class gets over or within this week, basically. So to summarize this lesson, you can see that chromosome slide preparations are made by dropping the harvest cell suspensions on the wet slide. Then you follow it by drying. Then you follow the proper banding techniques. So you're going to get so that you get a karyotype and the chromosomes that are stayed. And then you have microscopes that are present. Optimal slides should have well spread but complete metaphase with very less amount of overlapping of the chromosomes, and you don't get cytoplasmic much. And then you have different, the phase contrast microscope is used to view this particular images of the chromosomes. And microscopes are present, like, in the cytogenetic laboratory for checking out different kinds of cells, for checking out different kinds of chromosomes also. So you have the dissecting or the stereo microscopes used for illumination with the opaque specimens. You have magnification, which is basically, how big the image is, and uh, how much the image is magnified by a secondary lens system. And then you have different kinds of lenses which help in giving like, better quality of the image and better resolution with the help of focusing. Basically, different kinds of lens magnification has been given. Lens magnification is by the tube length and the focal length. So magnification is tube length by the focal length. And magnification takes place in two stages, that is in the objectives and in the eyepieces. So good objective lenses are quite important in a cytogenetics laboratory. And then they have given basically eyepieces with different kinds of magnification, such as 8x, 10 times, or 12.5x versions are there. Quality of the image in a microscope is based on brightness, resolution, contrast, and focus. So this is all about your like, what you call it, staining and slide preparation and your microscopy in like, techniques basically. Now you have an appendix over here with different kinds of definitions. You just can go through it, appendix A and B. So appendix A is a glossary of all the genetic terms that are there. So you can see what is an allele. They started with A to Z basically. So you can just have a look at it. And glossary B is, appendix B is all the glossary about the different kinds of malformations that are there. So genetic diseases again, or different kinds of issues as such. Now we will start with developmental biology, and we'll just have a look at the basic concepts of developmental biology over here. When it comes to that, you get to see that we are going to see the principal features of development, developmental patterns in metazoans, and then we are going to see the uh, development of coelom, what exactly is segmentation, formation of somites, changes in the developmental pattern of the diploblast, development in protostomes and deuterostomes of unicellular eukaryotes, control of the developmental morphogenesis in Establarea, and then the studies regarding that. So introduction, when it comes to that, you get to see that, developmental biology is something which actually studies about the development right from the genetic to the molecular events, which help in the control and development of all the features in the living organisms. So you have molecular biology, you have genetics and cell biology, all these techniques which are used for to study about the development of a biological organism. Again, when you get to see the roots of developmental biology, when it started, so you can get to see that, Roux was one person, Wilhelm Roux, who was like, who started with, what do you call it, he had believed that fertilized egg receive substances that represent different characteristics of the organism. So, and then he conducted experiments on frog eggs, and another embryologist by name Hans Driesch, what he did was he had taken sea urchin embryos and started experimenting on them. So instead of destroying one of the cells of the two-celled embryo, he separated the cells from each other. He separated both the two embryo cells, found that isolated cells at the 4-cell stage also develop normally. So each cell retains all the developmental potential of the zygote is what he had concluded. So conflict between these two opposing views of development had been settled in favor of the Driesch's interpretation. So Roux, since he had done all these embryos, and he had taken the chick embryo basically, and he had done all these experimentation, so he is considered as the father of experimental embryology. And in 1800s, again, role of development was not known much to everyone. I'm unable to actually close my window. That is the reason this, what do you call it, uh, particular slide share is there. What I'll do is I'm going to stop sharing. I'll try to share again. I have another slide. Let me share screen again. So Egyptians, they have given different kinds of, what do you call it, right from the starting onwards, who had contributed what. Aristotle also had studied embryos of the different organisms, wrote a book, and Galen, then Leonardo da Vinci also dissection of the human fetus, and the embryonic, William Harvey, then Marcello Malfigi. So all of these is before the 1900s, in fact. So we have Roux from Fritz Muller had also done, and then you have William Roux. He had explained the cell division. So he's known as the father of experimental biology for that matter. And then August Weismann had given the germplasm theory, and Hans Driesch. So, like I told you, he had taken the cells and the partial embryos, and he had seen, that they also produce the normal embryos that were half-sized. They had given totipotency and discovery of induction. So, this is something that is there. We'll come to that. Then Sutton and Boveri. So, Theodore Boveri had demonstrated normal development, which is dependent upon the normal combinations of chromosomes. So, and then you had Thomas Hunt Morgan, who had used Drosophila melanogaster, and he had suggested, or he had demonstrated that genes are located on the chromosomes. This helped to establish that the basic principles of transmission genetics. Then Thomas Hunt Morgan's student H.J. Muller demonstrated that ionizing radiation could be used to induce the mutations in the Drosophila. So a lot of work again based on on Drosophila had helped in the developmental biology. Now, if you see any kind of most of the developmental biology has been with the use of Drosophila melanogaster. So again, you have quite a number of important concepts that are coming over here. So, as to the specific role of the genetic material in development, how does the nucleus regulate the temporal and the spatial concentration in spatial coordination in cell differentiation, all of these. So, just give me a moment, please. So you have different scientists that have performed different kinds of experiments, such as Hans Spemann and his student Hilde Mangold. So the organizer effect was seen, and then of course discovery of the organizer effect and the embryonic development was given to Hans Spearman. Then you have induction, that is the coordination, the spatial coordination, excuse me, in the cells. So again, one group of cells which change the behavior of the adjacent group of cells, so that there is a change in the shape, mitotic rate, or the fate of that particular cell. All this is known as induction. So again, discovery of it has been done, and they removed the cells from the dorsal lip of an amphibian blastula. They put them in a different location altogether on another blastula. So what happens is the dorsal lip region of the mesodermal cells actually produces a notochord. So this resulted in the formation of two notochords. One normal dorsal notochord, second along the belly, because it was transplanted to the belly region. So they have given a picture of it also. So it was Spearman and Mangold who had given this. And then rise of genetics from experimental biology was done by Thomas Hunt Morgan. And then Morgan had seen that the hereditary determinants lay in the cytoplasm. But then they removed the cytoplasm from uncleaved eggs. You found that there were defective embryos formed. So there's a timeline which Wilson student and Morgan had found mutations in Drosophila, integrated or interpreted that it was because of the X chromosome. So you have the gene theory that actually comes out, which says that genes are physically linked on the chromosomes. So these were the basic concepts that were actually seen. So the principal features of the development are that development consists of both genotype and phenotype. Nuclear genes are not lost or mutated during the development. Genome of every cell is equivalent. Exceptions are the lymphocytes, because during differentiation of the lymphocytes, DNA is getting arranged to create immunoglobulins and the antigen receptor genes, which is based on the antigen that comes. So genomic equivalence is implied by metaplasia, in which one differentiated cell type becomes another differentiated cell type. So if at all a single cell fertilized egg give rise to different types of cells, that is muscle cells, nerve cells, this is the cell differentiation. So we are talking about, and that nuclei from the differentiated cells direct the development of the complete adult. So this is the principle of genomic evolution, and a small percentage of the genome is expressed in any kind of cell for that matter, and DNA is the one that gets transcribed. Different cells show different regions of DNA being transcribed. So you have northern blots or in situ hybridization and PCRs, which show the transcription of the particular genes. And functions of this gene can often be seen using the antisense mRNA or the transgenic expression of the genes, that is the gene knockouts. So when it comes to the developmental patterns in the metazoans, let us just have a look at it. So this is basically the life cycle of any organism for that matter. So you have fertilization, cleavage, leads to gastrulation, organogenesis, metamorphosis to gametogenesis. Once gametes are formed over here, then they get fertilized. Then again, the cycle goes on. So again, stages have given. Then when it comes to the developmental pattern in the metazoans, you get to see that the last common ancestor of the bilateral animals, that is the Urbilatria, was segmented. And then you see that segmentation may have undergone a lot of complex derivations in different kinds of phyla. So you have striking similarities in the mesodermal pattern in all the ancestors. And again, there's a diversity in the bilaterians as well from the metameric ancestor that is presented. So several mechanisms of this evolution have been seen, which operate on the segmented ancestral body plan. And this helps in the emergence of the different kinds of bodies right since the Cambrian period onwards. Now, there are a lot of advances that have been done regarding the phylogeny of metazoans and identification of the pattern genes basically, so that their morphology can be seen, and the last common ancestor of the bilateral animals and their body plan came into being. So you came to know about how things worked out a long time ago, and how what do you call it, the body plan of um, the evolution of body plan rather. So you have that. And at the same time, on the basis of this molecular phylogeny of metazoans, Urbilateria, which is basically the last common ancestor of the bilateral animals, that had been seen. So you have the metazoan phylogeny. This had given rise to a lot of new data. Now, last common ancestor of insects and vertebrates, that is the bilaterian animals, was the Urbilateria. So again, remarkable similarities were found in the development of the genetics, or the origin, or how you say, between fruit fly and the Urbilaterians. Ancestor of annelids and arthropods. Again, their respective metamerism, segmentation of the body, has been considered homologous by a lot of zoologists. It says that it is the ancestor of all the protostomes. So the ancestor of protostomes, excuse me.
Daddy got angry for.
Right. So again, you get to see that ancestor of annelids and arthropods is also the ancestor of protostomes. So if at all the ancestor of protostomes displayed a segmentation compatible to that of the annelids and arthropods, this means that segmentation was not lost completely, or it was not independent from that of the protostomic lineage. Now, if you see that metazoan phyla, and the most important fact is the presentation of the living fossils. Platyhelminths have a relatively simple body organization. They are considered to be a remnant from the flatworm-like ancestral bilaterians. So now they are you can tell that the flatworms have been derived from protozoans whose ancestors lost the coelom, they lost the anus, and they lost the circulatory system altogether. So you have a contradictory view to this with the molecular data, which says that 18S RNA and the myosin sequences that are present inside this, with that information regarding the 18S RNA and the myosin sequences, some authors find out that acelomorphs are a group of very simply organized flatworms, and they happen to be a potential sister group to the rest of the bilaterians altogether. So let us just have a look. Right. Now, when it comes to the coelom, that is the body cavity, let me see. I have some other basic concepts, having this kind of information, whatever has been given in your book, has been slightly difficult for me to obtain. I'm trying to see whether there is anything regarding what we are studying or what's been given in your book, but it's turned out to be slightly difficult, like I told. This is totally irrelevant. I'll just put this in. Now, when it comes to coelom or the body cavity, you get to see that coelom is the body cavity that is lined by the mesodermic epithelium. And according to the simple definition, that large, most of the bilateral animals can be called as coelomates. So the function of it and the structure of it, differs in different phyla and also within some phyla. So in annelids, you have a hydrostatic skeleton, while in mollusks, the coelom is reduced or it is present only in the cavity around the heart, gonads, and the kidneys. In the arthropods, the coelom is present during the embryogenesis, and it is entirely replaced in the adult stage by a hemocoel. In echinoderms, so again the coelom is in the form of a complex water canal. So you have acoelomates, you have pseudocoelomates, you have true coelomates. Nevertheless, whatever irrespective of the coelom in the adult stage, during the development you have a coelom that is present. Beside the coelomate taxa, you also have the pseudocoelomate groups, that is that's there in this body cavity. Nature varies considerably in the different kinds of phyla. Complete absence of body cavity in the enteroprocs and the gastrotrichs. They are known as the acoelomates. To the priopulate body cavity, which is known as the true coeloms, which is present in nematodes, nematomorphs, and chinorhynchs. So you have over here in the nematomorphs, chinorhynchs, and the nematodes, it is an embryonic blastocoel that is present. You have formation of coelom, enterocoelus, and the schizocele coelom in your textbook. You can just have a look at it. So it may not be homologous in the first place. And the development of the coelom can be divided into two ways, that is enterocoely and schizocele. Enterocoely is pouches forming from the archenteron wall, and schizocele following the with the mesodermal blocks in the protosomes. So according to this, different modes have been given over here, and there is a contradictory study regarding the homology of the coelom, in fact. Now, when it comes to segmentation, again, it is quite controversial as such as compared to the coelom definition as well. So what exactly is segmentation? What exactly is metamerism is something that you have to, it's not exactly clearly given. So an organism is considered metameric or segmented when its body is formed of repeated serially anatomical units, which is known as metameres or segments. So this definition can apply to annelids or arthropods. But in addition to other serially repeated structures, which is seen in the unsegmented bilaterians. So full metamerism would be much more significant phenomenon, and it requires a morphological, physiological, developmental adaptations in each segment, in each repeated segment for analysis. So again, this is flawed if a metameric organization of the body is lost at some point of time during development. For instance, in arthropods, there is a loss of extensive metamerism, but you still have remnants in the form of repeated organs, such as limbs in the cladocerans or spiders, or the heart ostia in the spider opistosoma. So if at all there is a loss of the segments, then you cannot exactly call it metamerism, or this particular organization cannot be defined as such. On the other hand, full metamerism has given rise to repeated organization of the organs, and this can still be seen in the living organisms right now. So the phenomenon of segmentation was considered in all the different kinds of manifestations. And again, there are two problems that came out. First is organs affected by segmentation. They are always not the same. So you have seriation, that is selective loss in some of the organs as well. the new organs can also start coming and adopt according to the general segmented pattern in the body. So you can see this in the axial skeleton of the vertebrates. So whether you classify the animals according to the segmentation or you cannot classify according to them is, considering only this one particular system of organs or one particular germ layer, you will have to check. And then you also have the seriation found in different organs. This can be discontinued from from two different organisms. So Amphioxus is one organism wherein there is no relationship between a seriated, that is serially placed gill slits, and the myotomes, muscle segments. In vertebrates, there is a disconnection between pharyngeal arches, somites, and brain segmentation. Again, this differentiation pattern appear independently. So you cannot exactly couple them all together. So you have to separate them and look at it during the developmental period. For example, anterior somites appear in good correspondence with pharyngeal arches during the embryogenesis of basal vertebrates such as the lampreys or sharks, while they do not correspond with the amniotes. Now you have this neural crest over here, which is a temporary group of cells which is present uniquely in the vertebrates. They give rise to the ectodermal cell layer. They give rise to diverse cell lineage. Another 10 minutes, I'll try to finish this lesson as much as possible. I'll stop with the protostomes and the deuterostomes. This can be continued tomorrow again. So, let me finish this particular somites, and then I can relieve you. So these ectodermal layers, the neural crest cells, give rise to the diverse cell lineage. As you can see, the melanocytes or the cartilage or the ganglia. And then this is how the neural plate and the neural tube occurs basically. Epidermis, convergence of the neural fold. This is the neural groove. This is the neural crest with the epidermal layer on the top, and this is the neural tube. So they have a multipotent capacity basically, and they have a migratory property and a neural crest cell, and the different kinds of regions that are divided. And you have, this is how it is: cardiac neural crest, aortic arch, basic developing of the arch. Now you have somites. So the mesoderm, what happens is, forms at the same time as the two germ layers, that is the ectoderm and the endoderm. So mesoderm at both the sides of the neural tube is known as the paroxyl mesoderm. So as this primitive streak which is present, it regresses, that is slowly becomes smaller. Neural folds gather, they become the tube. The paroxyl mesoderm separates into blocks called as somites. So these are the tube formation. So what exactly are somites? They are the blocks of mesoderms that are located on both the sides of the tube, the developing vertebrate embryo. Precursor populations of the cells. They help in giving rise to the different structures in the body and will eventually differentiate into the epidermis, dermis, cartilage, tendons, and vertebrae. So this is the somite formation. So this is again different. So, in different bilaterals. These are the somites. These are the presomites in the mesoderm. This is ectoderm. These are the somites. So, segmented presomatic mesoderm depends upon the segmentation talk established by cyclic, expression of the number of genes. So, we have the notch pathway, Wnt signaling pathways that are expressed in the oscillating pattern in the mesoderm basically. Again, notch protein activates the segment patterning genes that establish the somites, and they're regulated by combination of the RA and FG8 and Wnt signaling pathways. So in humans, you have around 42 to 44 pairs of somites that are present along the neural tube. Several caudal somites disappear again. That's why you have only 35 to 37 somite pairs which are getting counted. And number of somites and number of age related. So what happens is the fate of the somites considerably varies in the bilaterians. Annelids, the whole of the animal body cavity is derived from large extensions of the somatocele. All the mesodermal derivatives in the trunk, that is the somatics, planktonic muscles, septa, blood vessels, all of them come from the mesodermal lining. In arthropods and onychophorids, somatocele when present doesn't take a full extension. It finally disappears. This is replaced by the hemocele. So you have the somatic differentiation again. So again, in lancelets, the trunk somites extend fuse ventrally to give the unsegmented perivisceral coelom. Vertebrates, the mesoderm is divided early into several components and the dorsal somites do not contribute to the adult coelom. So, somites play a comparable role in the ontogenesis of seriated organs in distinct organisms. Either these structural units directly come from the mesodermal epithelium or their seriation is apparently induced by the somites. Organs that they produce include the segmental muscles, the nephridia, the gonads, and the gonoducts. In short, if you see, somites are the most extensively segmented animals. They are present in the three most divergent bilateral lineages, and they play similar roles in the organogenesis, that is, organ formation. Again, development of the metazoans, that is multicellular animals that pass through this embryonic stages of development, is present with a lot of illustration actually. So you can get to see that life has not come or evolved in a straight line as such, but there is a lot of branching that takes place during the evolutionary path. So you have metazoans belonging to diploblasts, protozoans, and the deuterostomes. So the metazoans belong to all the three major branches. Sponges develop in a manner so different that they are not considered as metazoans at all. They are known as parazoans. Sponges have three different types of somatic cells, and you have archaeocytes which can differentiate into any other cell. So they are say multipotent or totipotent cells. And individual cells of a sponge which is passed through the sieve, they can rearrange. You have regeneration or the formation of new sponge cells, and you have even reaggregation which is species specific in sponges. So sponges also have, they do not have any kind of mesoderm. So porifera do not have any kind of true organ system. They do not have a digestive system nor a circulatory system as well, nor you see nervous system or the muscular system, unlike the metazoans. So, but then they share different kinds of features of development, that is the gene regulatory proteins or signaling cascades, suggesting that they have a common origin as such. We will see about the diploblasts in the next class. I will have to stop right now over here and then we will check for the diploblasts. I will send this particular slide to you so that you can see and know the different kinds of definitions. What a myotome is, what a somite is, and what is the neural the crest, and what exactly is the neural different kinds of neural crests are there, the cells related to that. So when you start seeing that, you get an idea as such, how these help in the development. So I will stop right here. We will continue with tomorrow, the lesson tomorrow again. Thank you. I'll send it to your group. I think I've sent it some of your topics to the group basically. So I will just send it to your group. All right. Yeah. Thank you.